my name is Dania Hailat. I'm a student in Dr. Hancock's genetics lab here in the University of South Carolina-Aiken. And we're studying the effect of the nuclear export signal on harbinger transposition in human cells. So essentially, this research aims to make a mechanism used in gene discovery more efficient. So understanding the function of human genes is really important, especially for scientists when they want to develop drugs to help treat human diseases. Now, the problem is that more than 20% of human gene functions are unknown. And the way scientists are trying to tackle this problem is by mutating a gene, because if you can mutate a gene, you can gather clues to that gene's function. One way to mutate a gene is by using transposable elements. So we're researching a way to make transposable elements more efficient in mutating genes. Now, I've mentioned transposable elements and their role in gene discovery, but I haven't really told you guys what transposable elements are. So transposable elements are mobile DNA sequences found in almost all eukaryotes. And when I say mobile, I mean they can jump from one place in the DNA to the other. The way they work is they produce proteins that enter the nucleus, latch onto the sides of these transposable elements, pick them up, and insert them somewhere else within the DNA. This is called transposition or jumping. Now, as you can see, one of our transposable elements has landed here in a blue rectangle, signifying a gene. And when a transposable element inserts itself into a gene, that gene is inactivated or mutated. Inactivated or mutated genes can help us identify gene functions. So the specific transposable element that we are working with is called harbinger, and the reason we decided to use harbinger is because although harbinger is originally found in zebrafish, it has been shown to jump in human cells, meaning that we can observe its effects in human cells. So the goal is to increase the transposition frequency of harbinger in human cells. And the way for us to do this is first we have to understand the structure of harbinger. Now, harbinger encodes for two DNA sequences, TMP and midlight. Each of these sequences encode for a protein, or are responsible for producing a protein that enters the nucleus, latches onto the sides of harbinger, and allows it to jump. Now, one of our DNA sequences has a NES, here in the dark green, which stands for a nuclear export signal. And like the name suggests, this signals our TNP protein to exit the nucleus. Now, sometimes it signals it to exit the nucleus after TNP has already helped harbinger jump, and other times it signals it to exit the nucleus before TNP ever has the chance to help mobilize harbinger. So this leads me to our hypothesis, where we predicted that if we can get rid of this nuclear export signal, then we could allow for more time for TNP in the nucleus to help mobilize harbinger. Now, for us to test this hypothesis, we would have to measure and compare the transposition frequencies, or the transposition rates, of both our regular harbinger with its TNP that has its nuclear export signal, versus our mutated harbinger, which is our harbinger with its TNP but without its nuclear export signal, and this would be referred to as our TNP minus NES. This is our experimental. So first, here we have our TNP protein in a circular form called a plasmid, and this is our control. Now, we use this control to create our experimental, which is our harbinger TNP minus NES, which stands for without its nuclear export signal. Now, to create our experimental, we first used restriction digest enzymes, which act as scissors to cut out our TNP. This left us with a gap in the plasmid, and we filled this gap with a G block, which is just a synthesized piece of DNA of TNP minus NES. Now, using a process called infusion cloning, we were able to create a full plasmid of our experimental, or what we believed was our TNP minus NES. Now, to verify that this is indeed our TNP minus NES, we ran some tests. The first thing we did is we inserted it into bacteria, and from this bacteria we screened 13 of these colonies, and if our construct was our TNP minus NES indeed, then we would have expected plasmids at about 680 base pairs, and as you can see, we had multiple colonies that showed us this plasmid with our expected base pairs. So for further testing, we took these colonies in the green box and we ran a digest, and we also expected a specific plasmid at 5,000 here and about 1,000. And once again, multiple colonies or multiple plasmids had this band. So we proceeded with our plasmid in lane number 4. Now, before we ran our transposition assay, we decided to sequence this plasmid just to make sure that it was indeed our TNP minus NES and it was what we expected, and we sequenced it and it was indeed what we expected. So now we can move forward with our transposition assay. Now, our transposition assay, it's just a test that tells us how much transposition is happening. So we did this both for our control and for our experimental, and then once we got our transposition rates, we were able to compare them to each other and see which one had the highest transposition frequency. So I drew up a diagram of the process that we did. So here we have our Harbinger plasmid with our TNP and MIB-like plasmids. Now this is our control, and we know that because our TNP has its nuclear export signal. So what we did is we inserted these plasmids into a cell. Now naturally the plasmid, our Harbinger, it's either going to jump and incorporate itself into the DNA of the cell, or it's not going to jump and it's not going to incorporate itself into the cell of the DNA. Now, to quantify this and measure the transposition frequency of it, what we did is we inserted a drug called G418 that killed every single cell in which Harbinger did not insert itself into the DNA of the cell. And the way that this works is that Harbinger, it has a neomycin gene that is resistant to G418. So when it jumps into the DNA of the cell, it brings along that resistance to G418. So basically what we did is we tied every single transposition or jumping event into a living cell. So if it jumps, we have one living cell. That way, we correlated the culture that had the most amount of living cells is also the one that had the most amount of transposition. So like I said, we did this with our first condition, which was our control, Harbinger, TNP, and MIB-like. We repeated this transposition assay with Harbinger and MIB-like, but we exchanged TNP for TNP minus NES, and this served as our experimental. Now, we repeated this again without any TNP. This served as our negative control, and then one last time without either helper plasmids, which also served as our negative control. So, based off of the research that we did, we had expected results, and our expected results was to have the highest amount of transposition amongst our TNP minus NES, followed by our TNP, and then our lowest transposition rates we expected to come from our MIB only and our Harbinger only, which, like I said, served as our negative controls. Now, what we actually got, our actual results, were somewhat similar in that TNP minus NES appeared to have the greatest amount of transposition, so it appeared to have higher transposition than our TNP. However, what was not expected, as you can say, is that MIB-like, or MIB only, had a higher transposition rate than we expected. But this was not a statistically significant difference. In fact, the only statistically significant difference we had was between our TNP minus NES and our Harbinger. So, based off of these results, we were able to conclude that TNP minus NES induced more transposition than Harbinger only. Now, consistent with our hypothesis is that TNP minus NES appears to produce more transposition than TNP here in red. However, additional experiments are needed because it was not a statistically significant difference, so we cannot conclude it 100%. Now, if we were to repeat this experiment with more conclusive data, then this could provide a more efficient approach to mutating genes in human cells so that we can identify their functions. Thank you.